All right. Hi, everyone, and welcome. I'm Monica Daly, and I'm an associate professor in the Department of Ecology and Evolutionary Biology at UC Irvine. And I'm happy to welcome you to another workshop in our series on integrative organismal modeling of movement. These workshops have been sponsored by the National Science Foundation Division of Integrative Organismal Systems, with the goal of bringing together scientists at the intersection of biology and physics. We aim to highlight the use of model-based and mathematical tools to address fundamental questions in organismal biology. And we hope that these workshops will be accessible introductions and will help to generate dialogue and collaboration to help both experimental and theoretical scientists make effective use of mathematical and modeling tools for understanding organismal movement and behavior. The workshops will be recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences. So you can go there to see our archive of previous workshops as well. And with that, I'll turn things over to our speakers for today. And please welcome Matt McHenry from UC Irvine, Saad Bamla from Georgia Tech, Arvind Santhanakrishnan from Oklahoma State University, and Eric Titel from Tufts University. Thank you.
Thanks, Monica. So I'm going to give a brief introduction. Here are the names of the presenters, but also Laura Miller was our organizer. And if you haven't caught it yet, Laura gave a fantastic presentation on computational fluid dynamics approaches, which shows a great complement to today's presentation. So the agenda for our presentation today is as follows. I'm going to talk about flow visualization in general. I'll give a little bit of a historical background on PIV. Then Saad is going to give a live demonstration of PIV methodology. Arvind's going to explain some of the fundamental principles and the basis for methodology on PIV. And then Eric is going to follow up with a demonstration of some of the software that can be used to analyze flow fields and the post-processing of those flow fields. So fluid dynamics has fascinated naturalists and those interested in the physical sciences for centuries. Here we have an example of some qualitative flow visualization provided by Leonardo da Vinci. And his studies of fluid flow were of interest not just for aesthetic reasons for his paintings, but also he was interested in engineering and biomechanics as well. And so what we see here are some of his studies on the human heart. We have a heart up here, and this is showing vorticity around a valve, and he had hypotheses about the openings and closures of valves according to the vorticity within the chambers of the heart. Here we're sort of jumping forward into the Enlightenment era, and this handsome guy here is Osborne Reynolds, and this is a figure from his classic paper that introduced the Reynolds number. So this is an era where you'd include a self-portrait in the methods of one of your papers, but it's showing a really clever device that was used to arrive at the Reynolds number, which you may have heard of. What this is up here is a tank of water, and there's a funnel at one end, and that funnel's connected to a glass pipe, which is continuous with this pipe that comes out down here, and then there's a spigot here. So all the water in this tank spills all over his lab floor, and he can control the rate of flow with this lever up here, which controls the valve down below. And he can measure the drop in flow with this little device here. And to visualize the flow, he has a vial of ink here, which he injected at the opening of the funnel. And by monitoring what the flow looks like in that glass tube, he was able to arrive at the variables that control the appearance of that flow. And this was reconstructed in modern times. And so what we're seeing here are the walls of the glass pipe. And in the center is that stream of ink, which appears just as a straight line. And what he found is that by varying the diameter of the pipe, by varying the fluid flow through it, through that lever, and by changing the viscosity and the density of the water, he was able to, through any one of those variables, get this stream of dye to transition from a little line like that into something that looks like this. He could trip turbulence, and he realized that could occur with any one of those variables. And actually, if you could create a mathematical expression, a ratio of those quantities, then it doesn't really matter which one you vary, you can trip it to create the turbulence. In addition, if you increase the diameter, increase the flow, reduce the viscosity, or increase the density, then you can get that dye stream to be all the more messy and more turbulent like this. And so you have him arriving at this ratio, which can now be arrived at purely mathematically, but at the time it's an empirical result, this Reynolds number. And so the Reynolds number, of course, can characterize not just the nature of the flow, but you can infer a ratio of inertial to viscous forces. So what this illustrates is just the outset. The foundations of fluid dynamics are very much dependent on the ability to visualize the flow. It is a sort of fundamental aspects of empiricism in the foundations of fluid dynamics. I'm going to fast forward into 1930 here. Here we have Ludwig Prandtl, who devised this device here, which is a hand-driven flow tank. And he would make recordings with this camera up here of particles in suspension. And so this is a sort of early version of what we're doing, what we'll be demonstrating today. What we see on the left are some of those particles in suspension around an airfoil. This is before Ludwig Prandtl became a Nazi and contributed to the war effort in Germany in World War II. So he was maybe a sort of notorious figure at this point, but he did identify mechanisms for stall. So if you imagine airplane wings at very high angles of attack, they fail to maintain circulation because of this shedding of vorticity that we can see on the top surface, which creates a catastrophic drop in lift. There is a fantastic series that was supported by the National Science Foundation in the 60s, which I draw on a lot in my own classes in biomechanics, that illustrate a whole variety of flow visualization techniques that are largely qualitative, fantastically instructional, and also can be used as a basis for making measurements. So what we see here is a flat plate from one of these videos. And by the way, they're archived at MIT. This is a fantastic resource. And on the right are all these topics that are illustrated visually in these videos. Here's a flat plate. This technique is in a flow tank where there's a wire that when you run electrical current through it, it creates hydrogen bubbles. And by turning the electrical current on and off, you can create stripes of hydrogen bubbles in the fluid. And so what this is illustrating here is the boundary layer interaction with a flat plate over the surface. And they clearly spent a lot of time putting these movies together. And so for example, here, now they're moving the camera with one of these stripes of hydrogen bubbles, and you can see the development of the boundary layer over the surface of the plate. So this series is just like mesmerizing and fantastic. And so I highly recommend it if you're not familiar with it. None of it, I think, is particle image velocimetry, but it illustrates just the value of a variety of flow visualization techniques. Here's another one here, which is illustrating fluid dynamics at low Reynolds numbers. And so this is a glycerin solution contained within two concentric cylinders. So they're going to rotate the inner cylinder to illustrate the shearing that is generated by that motion, and also the viscous adhesion on the surface of that inner cylinder. And another cool thing about this video is it also illustrates kinematic reversibility. So they just sort of turn it around in the opposite direction, and at these low Reynolds numbers, you can exactly replicate the fluid flows in reverse. And they have another one where they write like RE less than 1 in the glycerin as well. Now, around this time, those with an interest in biology started to apply some of these techniques to looking at biofluid dynamics. So Rosen in 1959 is a classic study supported by the Navy, and it was testing this hypothesis that the fluid flow around a fish is perfectly laminar, including in its wake. And he devised a clever technique of adding milky water on the floor of the tank, and the fish is shown right there. And through the wake generated by the fish, it creates vertical mixing between that light milky layer and the darker expanse of fluid above it. And by allowing the fish to swim through that volume, he was able to create movies such as this. This is by far the most beautiful image from that book, but even the messy images allowed him to make measurements of the wake. And so it's not just purely a qualitative approach. And from those measurements, he was capable of inferring the fluid forces. These are more hypotheses than measurements of fluid forces. But at any rate, even by the 60s, we had this capacity to visualize flows and start to infer the mechanical implications of those flows. And I'm just jumping around here historically, by the way. There are lots of really important innovations and insights gained by fluid dynamics, but I'm just, in the interest of time, I'm giving you the abridged version. Here we have an example of where things were at in the biological literature by the mid-80s. So Jeff Spett, still currently at USC, did for his doctoral dissertation work with Rayner and Pennyquick, where they use soap bubbles filled with helium in order to understand the aerodynamics of flight. And so here we have on the right a little pigeon wing and the resulting sort of messiness in the wake of the pigeon as it achieved a downstroke. And by exposing these soap bubbles with a strobe light of known duration, then the streaks of light that they pass give you displacement measurements. And then from those displacements, you can infer the velocity. So for example, he slavishly traced the particle paths. And so this is particle image velocimetry before the digital era. And from this kind of messiness, so here we have a pigeon wing, he was capable of making velocity measurements that looked quite elegant and using that as a basis of testing ideas, models that Jeremy Rayner had developed for the aerodynamics of flight. In the mid-80s, from this paper, they were unable to explain the lift generation entirely. When they added up all the forces according to their model, they had a lift prediction that was less than the weight of the bird. But when Jeff revisited this topic in the early 2000s, again in JEB, using digital particle image velocimetry, he had a much more comprehensive measurement of the messy wake behind a bird. And from that, he was able to make similar measurements in the digital era. And those were comprehensive enough to have a more high resolution image of the wake of the birds. And from this, he was able to predict lift that exceeded the weight of the bird. And we have other examples of sort of revisiting earlier measurements, including even Leonardo da Vinci. And so here we have Maury Garibe built a scaled model of this situation that was studied by Leonardo da Vinci, and using his PIV methodology was able to actually replicate this phenomenon that was observed or speculated on by Leonardo back during the Renaissance. So to offer some more illustrations of the applications of PIV, I decided I was going to highlight the work of some of our speakers today. I did this without their permission, so I hope I'm not misrepresenting them, but Saad is an author on this really cool study on protists, which will very rapidly change their shape from this sort of relaxed, elongated form into this compressed form. And when this occurs, they observe that protists in the neighborhood surrounding the protists that did this would respond with kinematic changes. And PIV was included in their toolkit for interrogating this phenomenon. And here we have a liquid film that includes particles that allowed for measurements of PIV to find out that this rapid motion up here creates a disturbance in the flow surrounding the protist, which allows for hydrodynamic communication. Arvind uses PIV to study the flows around dynamically scaled models. And so what we have up here, he's interested in the metachronal waves, that is, shrimps and other crustaceans and their abdomens have a whole bunch of these paddle-like structures, and they coordinate the motion of those paddles to create propulsion. And so by building a physical scaled model at the same Reynolds numbers as the Chinese shrimp, by putting this in a viscous solution, they're able to replicate the hydrodynamics. And the great thing about a model is you can instrumented to measure force, you can also control its kinematics. And so here's an example of imposed kinematics. So you can consider the forces not just that are generated by the observed patterns, but you can vary the patterns to see how the observed behavior compares to what's theoretically possible. And here we see an example of some of Arvin's PIV measurements where we have two paddles shown together as a function of time as we move down this column. And we have measurements of vorticity that results from their motion. And from those measurements, he's able to infer the momentum flux at different Reynolds numbers. Reynolds numbers are another thing that are easy to manipulate with scaled models where you might not be able to get the animal to behave under different Reynolds numbers by varying the viscosity of the fluid. Eric Teitel has been educating me about fish propulsion for years. Here we have a beautiful measurement of a flow field from this study from his dissertation. He very nicely described the flow patterns generated by a swimming eel, but then could quantify the fluid forces and compare them to classic models of elongated body theory. So the traces here are Lighthill's elongated body theory models with his measurements from PIV shown in red. And that allowed for the formation of a model for how eels can generate propulsion, which really puzzled me for a long time about, well, the jets in the wake of an eel point sideways instead of backwards like they do in a bluegill sunfish. And Eric had to very patiently explain to me why this makes a lot of sense. So with that, I'm going to hand it over to Saad, who's going to do a live demonstration of PIV measurements.
Hey, Matt, how are you? What's up?
Good. How you doing?
Good. I was just walking around in the hallway. You want to see something cool?
Yeah.
Okay, let's see. I'm going to turn my camera around. Let's walk in the lab and let's see what's going on. All right. Are you guys ready? And you can hear me okay?
Yes.
So the lab is a little bit loud. So we'll speak loudly and you just give us some feedback on whether you can hear me or not. So
welcome to the lab. Victor here, Victor Ortega.
So Victor is a research scientist in the lab, and you've got a few demos for us to talk about.
Yeah,
PIV.
sure.
Okay, so let me switch
we
the camera.
need to turn off the lights.
Okay. And I might give you all some vertigo by moving around, but we'll give you a view of where the action is. So Victor,
take it away. Let me explain a little bit this setup. So we are working with small animals. But now I'm going to talk about flamingos. We are working with a project about flamingos, and flamingos, usually they feed backwards. When they are feeding on the surface of the water, they are just moving with a beak pointing downstream. So I'm going to demonstrate some of I was listening about your talk about electrolysis. So we are using electrolysis actually to see the flow. And let me show you how it looks. So here, the setup is we are using a power supply. And we have two electrodes. Finally, we are breaking the water in a small oxygen and hydrogen bubble.
Can I just comment here, Victor, of your very resourceful water supply, which...
yeah, this is a small...
yeah, you can see Victor is very, very creative and, resourceful. And I don't know how he's taking showers at home anymore. But that's how sometimes, lab goes.
Okay. So, and we have our expensive PIV. I'm just joking. It's just a pointer laser. You can see here is less than $20 or $30. And one of the ideas is that this small setup is not that expensive, and you can work with different animals. In this case, this is a 3D reconstruction of flamingo, very small scale. And this is just this way. Sorry, and here you can see the vortices that is producing the flamingo. So you can see these beautiful vortices that are produced, and depending on the speed, I will increase a little bit the speed of the flow, and you can see those beautiful. But one of the ideas is that we are trying to test that this curvature that flamingos have in the beak is because they are taking advantage of the turbulence in the recirculation zone that they are producing, and that's why they are feeding in this region, and they are feeding backwards. And that's one of the ideas. And you can see here in this video that I'm showing you some of the particle traces. You can see all this region of recirculation that they are taking. This is the part of the beak that is bended. You can see that they are taking advantage of this recirculation zone, and we are suspecting that they are taking all the food doing this. No, so at the end of the day, they are feeding as a normal ram feeder, like you can think in a whale or a shark whale, and they are using just a small trick of fluid dynamics.
Okay, so Victor, tell us a little bit about what is that structure that is generating the black thing that I see? What is it? Let me see a side view. Can you describe how you made this structure?
Yeah, this is a 3D reconstruction of the flamingo head, and we use a photogrammetry technique, just taking several pictures of the beak, and we reconstructed that head. And you can see this the reconstruction. No? It's just a 3D printing, and that's why we are just mimicking the speeds at the flamingo is feeding and the structures. So we are studying using the PIV. But one of the secrets of this electrolysis is not useful just for the flamingos and 3D as a classical inanimated matter. We put also water striders in there.
Oh, wait a second.
Yeah,
can you give us a demo?
Yeah. And we can maybe illuminate with a light.
Look at that.
So these are different water striders that the hairy death, that those that jump. These guys are Ragobelia, and these are found in rivers here around Georgia. And you can see that they are fast. No, they require to be fast, because if not, in rivers, you need to deal with all this perturbation in the water. So let me show you what this technique of fluid dynamics, that this electrolysis works. So I have a video. It's hard to see in here, but you can see in here this video how the vortices looks like when you are using electrolysis. You can see the beauty of these waves that they are producing. They are actually flapping with these small wings they have at the tip of the legs, and that's the secret. And you can see here they look like some kind of jets, no, like the airplanes that they are moving around there. But this is one of the demos I want to show you with electrolysis.
One second. So can you say a few words about how big these systems are, just so we can have a sense
these animals are 3 millimeters in size.
And this is the same setup that you have here, and you just place the organisms?
Yeah, it's the same setup. Yeah, and one of the things I want to say is that why we are using electrolysis is because if you are using different particles, they can affect the animals when they are moving on the surface. And when you are using just these bubbles, no, that can be oxygen or hydrogen, they are not affecting the properties of the water, and they are moving as usual. You know? That's the good things about electrolysis. And we are actually working on a paper just to describe this technique applied to small animals. Okay. But let me show you other
what else you got?
So for the students, usually some animals, for example, as you was mentioning, that for birds, they see the environment with olive oil or small, like a bubble, soap bubbles, no? That they can put hydrogen, or they are neutral. But some animals are so tiny that are affected by these particles, are too big. So we are using just, you can see here, this is—
What is that?
It's just, you know, this for producing just—
Do you think I can
just
zoom
water
in?
droplets.
Oh, it's a humidifier from Amazon.
Yeah, that's a humidifier, and you can see here. Let me turn off this.
And this is another laser of yours from Amazon.
Yeah, and you can see here. So this is, you can see that it's very turbulent, right? It's very turbulent because, but let me put it in the other region, and you'll see this laminarity. So
let's hold on for a second. Let's try to see it. So maybe you can describe what this black structure is, so we can understand what.
So all these things are the water particles passing through, and because we are working with the small nematodes that they are just 1 millimeter in size, because we are thinking that they are dispersing by the wind. So we are putting the nematodes there. They are jumping, and we are using the PIV to describe the flow. No, that we are testing this nematode.
Hey,
so,
Matt, can you see this? What?
And you can see there.
Yes.
and then it's very laminar. So that's why this and everything is less than $50. No? And now let me show you something interesting.
Look at this.
So this is with a real animal, and this is
Courtesy of
courtesy of Jacob Harrison from Duke University. You can see
it's a little bit bright.
This is different because we are using this. These are big enough, and we are using just plastic particles. It's like a dust, no, that is used for fish or animals that size. So these are big enough to resist those particles. And one interesting.
1 second, hold on before you go. You see how bright
the settings of the camera.
1 second, yeah. Matt, is it visible or is it a bit overexposed?
Maybe something.
That's better. That's better. It was saturated, but now it's not.
You can see here that you can actually use this technique. You can hear them because they produce with a claw this cavitation bubble. So we are trying to do something, trying to understand the hydrodynamics of these cavitation bubbles with Jacob Harrison. And I think we have a very cool video actually that when the implosion of the bubble is occurring, it's producing a very beautiful turbulence at higher speed exactly after the bubble collapse. So we are trying to understand what is happening at these levels, no, with this very cheap PIV, and just the lesson here is that biologists can use all these techniques that are not that complicated that nowadays, because there is a free software that you can analyze all this. Just the only restriction is the creativity you have, no? Because you can see all these organisms can be studied in terms of hydrodynamics, not just kinematics.
Victor, can we do one more knock? I want to hear it. And you don't speak, okay? We want to hear that clearly and see if you can catch it. That was the sound of cavitation, right?
Yeah, that's cavitation. Yeah.
Okay.
Okay.
Thank you. So this is Victor. He's a research scientist in the lab. And now I'm going to walk over to Johnny. Hey, Johnny. Johnny's a PhD student in the lab. Tell us what you're working on.
Yes, so I'm working on a mechanical setup that I've built to try to mimic the fluid flow that we see in one of our other biological systems. And so in my project, I study another type of water strider.
Could we actually see the water strider?
Yes.
I see a video here, but actually let's see that actual organism to appreciate its size. Okay, we follow Johnny.
So as you can see, if you get in really closely, you can see some microvillia, which is a type of water striders that I have, moving across the surface of the water.
Oh, there you go. There you see a couple. Matt, can you see one?
Yeah, yeah.
So these,
there you go. That's
these specimens are only about 2 to 3 millimeters in size, and they basically walk on the surface of the water just as an ant would on land.
Why are you interested in this, Johnny?
So I'm interested in studying this specimen because this specimen overall is able to walk on the surface of water and land.
We're losing the audio now.
Uh-oh. Well, is it back again?
We can hear you.
We can hear you, but not.
Yeah.
Yeah.
Okay, Johnny.
So the reason why I'm interested in this specimen is ultimately because the microvillia has the ability to walk about equally as well on land as on water without really adjusting its gait. So in this video here, we can see how the microvillia is moving across the surface of the water and producing these beautiful counter rotating vortices. And so ultimately, using this mechanical setup, I want to understand how the fluid flow at these different speeds at which the microvillia can operate at.
Describe a little bit here what's going on in these two animated but very confusing two rotating pieces of wire. What's going on here? And come to my right-hand side.
Yes. So ultimately, what I did to create the system is that I created this basin of water here. And then these particles at the top are lycopodium, which is just dried plant spores.
What is the size of this?
They're about 50 microns in size. And so the reason why this water is green is because I also added some dye in order to improve the contrast for imaging and analysis. And so these motors are basically these stepper motors that I'm controlling through an Arduino. And then using this Arduino code, I can ultimately change the speed. And then from changing the speed, I can see how this fluid flow ultimately changes and to better understand like the different speeds at which these microvilli operate. Because with this system, I can operate at whatever speed I can and get a better analysis rather than relying on the microvilli to see at what speeds it can operate on.
Matt, what do you think?
It's fantastic. Yeah, really cool stuff. And I love that basically everything you've shown us could be ordered on Amazon and set up pretty quickly, it looks like. I haven't seen a contemporary use of the electrolysis like you're using there, and it's just fantastic.
Well, yeah, that is this gentleman's creativity, as you see, that he doesn't leave even a shower head, waste. He will bring some use to it. And really, that's one of the innovative elements, as you described. Matt, can you still hear me?
Yeah.
That he's taking all these classical techniques and applying it to invertebrates. And yeah, at these small scales, we can look at a lot of bulk and interfacial dynamics. So thank you. Let me just quickly thank Victor for his time.
Thank you, guys.
Thank Johnny. And of course, we are here for questions, and I'll switch the camera around and mute myself. And yeah, hand it over to the next person.
Well, Eric had a question.
Yeah, just a question and sort of a comment. Just your last setup there wasn't a laser, right? So how do you do PIV without a laser? Can you hear me?
We caught only a part. Well, it broke up a little bit. Do you mind repeating it?
I was saying in your last setup there didn't seem to be a laser. So how do you do PIV without a laser?
Well, actually, luckily, like because of the contrast, I actually still get very good lighting. And so I have done some analysis and some videos, and I'm actually able to detect the particle motion very well. I think it's partly because I'm just operating at the interface and not the bulk. But as of now, I'm actually getting good tracking of the particles, even with just this lighting. But ultimately, yes, a laser would be better.
Thank you.
And also, I just wanted to add one word that the organism stuff is actually done with a laser, the one you see on the screen. This one is still work in progress, and it still gives us some data. So, we can add in the laser to get really good stuff. But the advantage here is it's all at the interface, as Johnny just said, and you can appreciate that if I look a little bit through the side, you see that they don't actually come into the Z depth at all. They're just caressing the surface and creating these vortices. Are you asking me?
Oh, now I'm okay. Yeah, I couldn't for just about a minute or less.
Sorry,
I couldn't hear anything.
But yeah, that's all there is from us. Unless you guys have questions, happy to hand it over. But thanks for coming on our tour in the lab.
Thank you. Thanks to everybody in your lab. They've got a lot of cool stuff going on. Awesome. So I think I'm next. So Matt, I can probably jump ahead. So I'll probably, since Saad's got us all pumped up with all these beautiful real experiments and applications, I'm going to kind of do a little segue, talk a little bit about the basics of PIV, and then I was going to try to show a little bit of a demonstration of a processing program that is implemented in MATLAB. Can you all see my slideshow, or is it in the weird presenter view?
We see the presenter view right now.
Okay, let me swap the displays. Is that better?
Yes.
Okay, perfect. All right, so let me get started. And if anybody has any questions, feel free to put it in the chat, raising hands. Happy to. This could be a little bit on the drier side compared to the more live action filming that Saad had done just minutes before. But the basics of PIV, so it's again the abbreviation everybody here probably is familiar, is particle image velocimetry. So it's I guess the most sort of powerful feature of PIV is that it's not intrusive. So when we're trying to do, for example, biomechanics studies, we're wanting to try to probe the flow field, we don't want to sort of create artifacts just from our measurement. So that sort of affects the results. So PIV is really beneficial because it allows you to kind of use optical methods to visualize flow fields, right? And it's a whole field technique. So oftentimes you get a lot of data that you can use for years to come, because there's quite a bit of information in a whole flow field. And sometimes there's not enough time and bandwidth to even squeeze the data in a single paper. So, and the basic idea is that we're tracking movement patterns of these particles, and these could be a variety of different particles of different sizes and so forth. And we're trying to track the movement patterns between pairs of images. So this is an example of a classical sort of two-dimensional PIV setup where we've got a light source like a laser. We take the beam, we make it into a sheet, like this conical region sheet. And here we have some sort of jet going from bottom to top. And then we use a digital camera. And there's some sort of particles embedded in the flow that follow the flow and don't disturb the flow as a result of their presence. And then we get these images of what black and white sort of star lit night type of images. And then if you look at these little yellow boxes, we window it up into little bitty regions, and then we try to correlate the windows, each window between image pairs. And so then we get this velocity vector field, which every point here gives you a value of velocity in x-axis and y-axis, for instance. And this could be done over time. So you get, time varying sort of flow characteristics from this methodology. And so what I'm going to do next is to kind of go into the basics of like the seeding particles, the hardware, and those things, and then kind of talk about, the types of arrangements to get the different light sheets and so forth. And so the seeding particles, the one main assumption is that before the flow starts, because say this insect is flapping its wings or whatnot, you're assuming that the seeding particles are homogeneously distributed in the fluid medium. So there's not like some sort of patchwork sort of distribution or something like that. And the other assumption that's very sacred, I would say, is that the seeding particles faithfully follow the flow. So they themselves don't create their own sort of flow fields. And so which, like Saad showed, can be quite challenging when the scale of the measurement becomes smaller and smaller. Sometimes these particles themselves can distort the flow. And so the experimentalist has to make some really careful choices. And if you think about what the seeding particles, how we decide what to use, ideally you would prefer them to be neutrally buoyant in the fluid medium because you don't want them to sink something heavier. So they create their own sort of velocity gradient by sinking or floating for that matter. So if you use something like styrofoam particles in water, this is going to be a problem where they'll start to float. And so then your measurement is going to be impacted by the particle choice itself. And they're not supposed to be reacting with the fluid medium or the object. So in other words, these particles are going to be, they're going to have to be very inert to the fluid medium, and also any sort of, they can't sort of degrade the object, the test specimen, or anything like that. And so these are very important, sort of very basic seeding considerations. And the two sort of common types of scattering we see, one is called Mies scattering. I'm not going to get into like all these nitty-gritty details, but the essence of Mies scattering is that the particle surface area is what's going to tell how much light intensity you get from the particle. And this is, since we're using the images of these particles, it's kind of important that we try to optimize this light intensity. And so this can be impacted by the wavelength of the light. So maybe we're using bright light, maybe we're using a green laser, a red laser, what have you. And the refractive indices of the particle relative to the fluid medium. So there's a lot of different things that can make the scattering more effective or less effective. And this is fairly common. A lot of the sort of very classical PIV advancements, studies, they've been done using this sort of scattering idea. And so the common example I would say is like using theater sort of fog in air and wind tunnels and so forth, like Matt showed earlier with Jeff Spedding's work, or hollow glass spheres in water. So these are very common examples of seeding particles that employ the surface area based scattering. The other one that's sort of more, I guess, selective and gives you a higher signal to noise ratio is using fluorescent properties of the particles. So the idea is that you have specialized particles that absorb a wavelength of the light that's being used to illuminate the flow. And then they get excited and then they go to a higher state, they come back down, they emit some different wavelength. So then you can sort of optically knock out and just recover the emitted signal by using a lens filter. So in my lab, we use things like infrared lasers because we don't want to affect sort of visual perception by animals, for example, that are swimming. And so then we can use particles that are selective to a particular wavelength. We can use filters to kind of just get the emitted signal. And so the signal to noise ratio is higher when using fluorescent scattering because you sort of really selectively get the particle emission signal only. You don't get the background, noise and all of that. But one of the limitations is typically just limited to water. And an common example is a rhodamine dye that's used for these sorts of fluorescent scattering applications. And mainly because its particles have a high density, okay? And so the importance of the seeding particles is that the probability of finding a valid velocity vector. So now we've got really fast processing capability, even in our phones, for a matter of fact, right now. So it's not necessarily the processing that really kind of challenges the ability to get good data. It's that the raw data itself has to be quite high quality, and the seeding densities are very important criteria there. And so typically high seeding densities are preferred than low seeding densities. So in low seeding densities, so this used to be a very classical approach that's sort of being revisited now with more powerful algorithms called PTV, or particle tracking velocimetry. So what you see here is you have a low number of particles in an image, and you track the positions of those particles, the displacements in time, and you get the velocity from the differentiation of that displacement in time. But in PIV, we typically prefer higher seeding densities. And so, we're not interested in tracking individual particles. Rather, we're tracking patterns or groups of particles and not necessarily each particle with any sort of high fidelity. And so here's an example of classic sort of PTV on the left and PIV on the right. And you can see that the PIV gives you a lot more data. So the density of velocity vectors is more in the right-hand side image compared to the left-hand side image. And possibly the left-hand side, the processing is far more intensive because you're trying to label particles and faithfully follow them. The right is more sort of statistical, really, the outcome. So it's computationally more expedient, and you potentially can generate more data. And to kind of revisit the hardware, we think of like a digital camera. Laser is commonly used for a lot of the PIV applications, though there's been a lot of work lately with using bright light type of PIV studies. So the lighting is becoming far less of a restriction. Lasers can be expensive, especially very high power ones that you need for larger scale flows, like flow behind birds and stuff, things like that, larger birds and whatnot. And then the optics, generally for 2D PIV, we want to visualize just flow in a plane. So we want to take the beam of light and we convert it using a series of lenses to a sheet. And then there's some sort of controller a lot of times that synchronizes the capture of images with the light, and the system dynamics, for instance, if there's some sort of time scale with a wing bead or something like that. And then the software, typically we need some sort of image acquisition software. This could be open source, commercial. There's lots of different options out there, but they're called frame grabbers. A lot of the high-end sort of cameras, the high-speed cameras, come with their own software. You pay a lot for the camera, but they've got their own software for the frame grabbing, where they have lots of different controls you can play around with for exposure and shutter speed and all that. And then the other piece of the software is the processing. So there's cross correlation, which we'll talk about very soon. And usually the reason lasers were preferred is because PIV tended to be started in larger fields of sort of imaging, larger fields of view. And so lasers provided a lot more sort of coherent or single wavelength, high power illumination source. And that's possibly the reason why lasers were preferred when PIV sort of started getting more and more widespread use. And the lasers typically used in PIV can be pulsed or continuous. And the pulsed is like the word suggests, you've got twin sort of pulses, two laser heads potentially that come and pulse, and the continuous is just a beam that just shoots out. And then the camera selection, this is another sort of things to think about if you're trying to come up with your own sort of PIV system to study a problem. you want to think about the velocity range of the flow. And by that, the range truly, so like, you could have wakes in the flow behind the body of an animal, for instance, where the velocity is likely going to be slowed down. And maybe that's in an order of a few centimeters per second. But then the sort of the large, there's a much more higher speed flow sort of superposed around it. So there's quite a bit of dynamic range. So we want to think about what sort of cameras can be useful for that, for sort of acquiring the different time scales. So the low speed wake and the high speed sort of maybe environmental flow and so forth. And then we want to think about for the lenses, what sort of magnification we want, what sort of field of view we want. And really, ultimately, we want to think about how many pixels can I get per unit of length, like millimeters. So if I want to visualize flow in a copepod, I want to try to build up, a lot of pixels, for example, in a 2mm copepod or something like that. And then the camera resolution, typically we think about the pixel resolution, so how many are megapixels and stuff like that. And then the pixel size itself is important. So like a lot of the high-speed cameras that are cheaper, they tend to have large pixels, and so there's sort of some sort of averaging going on at the level of pixel, whereas the more higher sensitivity cameras tend to have smaller pixels. So that's another sort of criterion to think about. And then this dynamic range of the camera is basically what they call a bit depth. So they think about 12 bits, 14-bit camera. The idea is that we can quantify the amount of light into 2 power n levels. So you sort of discretize light, the signal that comes into a picture, in sort of these counts. And so how many counts your camera can discretize is basically, or sort of discriminate, is 2 power n. So 10-bit camera can only do 2 power 10 levels of sort of, or counts of discretizing that signal, whereas a 14-bit is going to go up as 2 power 14. And then the final thing that people look at is what's called quantum efficiency. So the wavelength and the x-axis here, the quantum efficiency is sort of like how sensitive is this camera pixel to the different wavelengths. And so perhaps for an application where you're using infrared, near infrared, like 808 or something like that nanometers, maybe you want to pick up a camera that has a higher quantum efficiency. And so depending on the light source, you want to think about what camera would be the most optimal for it. And the different types of cameras tend to be, more commonly is the Cmos camera. It's not as expensive. This basically allowed us to do slow motion or high speed capture in the present day. They have larger pixel size. CCD cameras are not as common, but they give you a lot more, quality data signal. They're more expensive typically because of their high sensitivity. And then in my lab, we also have these what's called SCMOS, scientific CMOS cameras, which sort of build a middle ground between the sensitivity and also the price factor. And then the beam optics. So typically your laser beam is two problems. It's usually the beam diameter is kind of big, and it's also the power of the beam is sort of not necessarily uniform. It's sort of kind of very rough if you think about the power as a function of the radius of the beam in different positions of the beam. And so what we want for a two-dimensional planar imaging is a very thin sheet, because we don't want particles that are going into the plane and out of the plane. We really want to just keep track of the particles as much as we can that are in that plane, where we're focusing, where the laser sheet is being subject in the flow field. And so typically the combination of lenses we use is things like a diverging lens shown here in the side view, and the top view is shown here. And so this kind of removes some of those high power, like sort of areas of the laser beam. So like try to kind of minimize the risk to burning out something in your setup because of the high power of the laser in particular regions. So this diverges the beam, and then you couple that with the converging lens. And then the most sort of important piece at the end is what's called a cylindrical lens. Sort of looks like a half round profile, like shown in the top. And so this basically spreads the beam into a sort of this conical region laser sheet. And so now we can sort of focus in on the particles in that sheet. And by varying the distance between these first two lenses, the converging and diverging lenses, we can make the sheet a little bit thicker or thinner. Sometimes people tend to thicken laser sheets a little bit for high-speed turbulent flows, because there's bound to be a lot more three-dimensional motion. So maybe you want to try to get a little bit more information without losing, just because the flow is highly three-dimensional. And then the two modes of PIV that are fairly common are what's called time series or high speed or time resolved PIV. And this tends to be particularly useful for very high speed phenomena like the cavitation, exclusion in the pistol shrink, that problem, for instance. You want to necessarily use a very high speed potentially for getting a lot of the finer details of the flow field. And so there, what we're doing is essentially the camera is exposed once and the laser pulse is drawn like a line, because the laser pulse is usually, if it's a pulse laser, it's usually just few nanoseconds. It's a very, very, very short pulse duration. If it's a continuous laser, then it's just sort of uniform and extends all the time. But this is for a pulse laser, and so the camera is open for a certain amount of time, and so then you acquire a picture there, and then after some time dt here, you acquire a second picture, and so on and so forth. Now the time separation between two pictures is usually 1 over the frequency of the recording rate of the camera. And so it's, typically you try to kind of look at how much particle displacement is occurring, and I'll talk about it very soon, to sort of play around and try to sort of optimize this recording rate of the camera so you get the most useful data that has higher cross correlation values. And then the other mode that is often used for very high speed flows where potentially the camera frame rate is an impediment, you cannot go really fast in a very high speed turbulent flow, for example, like some sort of hypersonic situation or something like that. Then what you do is you use these cameras that can acquire 2 images in a very short time interval. I'm talking like microseconds, several hundred nanoseconds, very few microseconds. And then to acquire another pair of images, they have to wait a little bit longer. So like, 1 over 15 seconds or something like that. So it's not high speed, but what happens is that you acquire an image pair in very short duration. And so the time between the 2 images of an image pair, and again, that's the really important part of the processing, is you're comparing 2 images, really. And so the 2 images, the time separation can be controlled by timing the laser pulse. So like one laser can start and then the other laser can be brought to fire really close to the first one. And so that timing thus is independent of how slow the camera can acquire one pair to the next pair, which can be, like I said, 15 pairs a second is maybe the rate limit for that camera. But in that one image pair, it can acquire images that are separated by 100 nanoseconds or something like that. So this is useful in high speed flows because you may not have cameras that can give you adequate amount of field of view, resolution, spatial resolution, and the sort of the high frame rates. And memory is always a problem. You don't end up having a lot of onboard memory in cameras, that your sequences sometimes are just a few seconds long. So maybe these can give you ways to get a long time sequence of a flow phenomena without needing a very, sort of high-end, high-speed camera. So if you think about sort of 3 different pictures that are shown here,
Do you add a comment that connects your stuff with what I was showing, just for the audience members who might be listening?
Sorry?
Can I add a thought here?
Oh, yeah, definitely. Go ahead.
I really like how you summarize, and I just wanted to throw in for the audience members that although we showed all the low cost stuff, which is good for very early stuff, what we are not showing is the high speed cameras, like you pointed out, which are probably a couple of 100K cords. And also that although the pen lasers start us off, really for what you're describing, at the end of the day, we do use high-end lasers. We didn't show them in the demo because we'd all have to wear goggles and put on the lab curtains.
Right. So
I think, just wanted to make the distinction to get some of the data we showed you on the computer screens. It does require second layer of really sophisticated stuff, but you can always get started. But to get quantitative and reproducible, very crisp data,
Right.
Any case, I just thought I'd chime in.
That's a good point. And I think, it comes down to the problem you're wanting to study, really, at the end of the day. If you design your research program around a particular startup budget or something like that, so then you can sort of go in the sort of the field of all the possible problems you're thinking of investigating, and sort of pick the best pieces of equipment. That's really where this goes, right? So If you have a lot of budget, great, get very high-end stuff. But if not, then maybe you want to think about what's the best money can get you at the same time, as studying the problem that you want to study. So the particle tracking tends to be on the top sort of picture. So you see a very clear particle there, maybe some defocused particles. So you can identify a single particle with very high accuracy. There's no uncertainty really as far as identifying a particle, but how much information you can get from that little picture is very low. It's just one particle. But then there's the opposite sort of bookend of the particle images is shown in the middle, and this is called speckled mode. And this used to be employed before PIV sort of became more popular in the 90s. And so here, the particle density is enormous. And so you can't really identify a single particle. It sort of looks like a little bit more along the lines of a white noise in a TV. You can't really sort of identify a lot of patterns, visually at least. And so it's very high information density, and it doesn't really visually tell you a lot of details. And like Saad showed, PIV data, you can make sense of it visually. You don't need to even process data to make, to get some of the basic qualitative observations. A lot of times you can sort of look at the data, you can see where there's vortices and uniform flow and, sort of all these different, distinguishing features you can find out just from observation. And that's because the sort of the optimal mode for PIV is high enough image density where you can identify single particles, but if you wanted to track every particle, from frame 1 to the last frame, it's just going to be computationally just very prohibitive. And so you don't necessarily want to do that, but visually you can see these particles and you can sort of kind of visually discriminate them is maybe the way to say it. And so if we sort of go into a little bit of the processing, sort of the nuts and bolts of PIV, here's an image of flow past the square block. And so what PIV is, again, let me re-emphasize, is that it's not this sort of, particle to particle to particle sort of tracking. It's more what we're trying to do is draw windows, right, in the raw image of these particles. And then in these windows, we try to compare two images and we see where the likelihood of finding a similar pattern between, let's say, the top left window of image 1 and the top left window of image 2. Where is that pattern most likely repeated? How much is that displacement? And that's really what I'm trying to figure out. So it's more of a windowed approach to the, we're really wanting to know what's the average particle doing in a window, and that's it. We're not trying to figure out what all these little bitty particles are doing in every window, because that's just computationally very infeasible. And to kind of go back, you can visually see what's going on because the particles are not displacing like crazy. So if can you guys see my mouse at all?
Yes, we can see it.
Okay, so if you look at that, where my mouse is at, and I'm going to straddle to the next frame, those particles are still there. They haven't left the window. They're still there, right? Some particles may have disappeared. Some particles may have shown up. Some particles maybe are going up. Some particles are going to the right. There's lots of different things happening in here. But you know what? We don't care about, the number of particles has to be the same. None of that. We're really interested in what's that average particle pattern look like in that window, and where is that particle pattern in the next window? What's that sort of lengthwise displacement? That's it. Okay. And so most of what we've been able to do from PIV started maybe in the 90s because of this digital camera revolution. And I say that with a little bit of, smile here, because this paper has been one of the most cited papers for DPIV, or digital particle image velocimetry. And Maury Garib, who's now at Caltech, and a student at the time, Christian Willert, who's now in the German Aerospace Agency, they pioneered this method, and they used something like, I believe it was like a 640 by 480 camera to show that this method works. And now I'm sure Saad, I know I do, Saad, we all have these multi-megapixel cameras, but the methodology scales. It works if your camera is really inferior in the pixel resolution or really, really superior. It doesn't matter, because the methodology is very much, identical across the board. And so this paper is where that sort of started. And so, I'm not going to get into the equations or anything, so I don't want people to get too hung up on it. But if you think about, let's say, an image in the bottom plate here, G1, so I've got some sort of X number of pixels by Y number of pixels in that image. And then in the top, after some time, delta T, there's another image, G2, right? So, and then these are the ones I'm going to try to compare, right? And that's really the heart of the processing here. And so what we do is we take the intensities of these particles. And so that's kind of what I'm describing here in the uppercase, the lowercase g. One is in the image coordinates, one is in the physical coordinates. But again, that's the math part. No need to get too hung up on it. But really what we're trying to do is to take one of those windows, think of it as a template. We're trying to kind of raster it around, so I'm taking this m by n window, and then I'm going to try to move it around in the second image. Literally, I'm going to prescribe move by 1 pixel to the north, 1 pixel to the south, something like that, right? So I'm going to move all of that. And at every position, I'm going to try to calculate the sort of how similar it is to that window in the original or the first image. And so very, basic idea would be to take the difference between the two, do some sort of least squares. You can keep the square root, take the square root off, but the idea is that you will find sort of the place where there's minimum, the shift, the m and n shift here, the m by n, the lowercase m by n shift, where you get the minimum sort of this quadratic difference, right? So that's a very simple way to think about it. So essentially, between image 1 and image 2, I can argue that it's shifted 10 pixels or something down and 10 pixels or something to the right, something like that, right? A more common implementation that we see nowadays is, so if you think about this minimum quadratic difference, you expand this and do like a binomial expansion, and then we get this, basically this cross correlation function phi, which really what it's trying to do is compare the first image intensity, that sort of G1, with the second image G2 with this m and n sort of shift. That's the pixel shift, how much displacement has been shifted. And so then when you plot this, you sort of get these, it's sort of these peaks and valleys and, little canyons and all of that. And so we want to land on the sweet spot where the correlation peak is the highest, right? And so if we were to do this, and this is something we can code up, in Python or MATLAB or whatnot, with the digital images in your phone even. But if we to sort of look at the amount of math this takes, so if I do a window that's like 32 pixels by 32 pixels, so one of these little windows is 32 by 32 pixels. I know it's not square, but let's imagine it's a square. Then this number of computations scales is 32 power 4. So that's like 1 million operations for one window. And now you can appreciate there's a lot of windows. So this is going to take a while, right? So this is where Willett and Greed, the paper I showed, they tried to use this sort of operation in not the physical space, but the Fourier space, the wave number space, 1 over physical space. And in a nutshell, this is a statistical result. But what they did was they took the fast Fourier transform. In MATLAB, this is like 1 line of code. So you take the fast Fourier transform of the first image, that window, the intensity, and then you get this G1 caret, and then G2, similarly, you take the fast Fourier transform of that, and then you take the complex conjugate of the second, and then, what you do is you do this sort of convolution of the two, and then take the inverse Fourier transform, and you get the cross correlation function like this in the graph, in the blue sort of meshy graph in the bottom. And so the number of operations is orders of magnitude smaller. It scales as n squared log to the base 2n. And so this allows you to actually do real-time calculations without having to wait for days on end for 2D PIV. So you can literally take these images, run them in a MATLAB or Python code, get results in minutes. And that's how quick the operation scaling is. And so a lot of these details on one of the first and still remains to be the best authoritative book on PIV is this by Raffel and others from the German Aerospace Agency. And there's also Steve Worley, who's a really micro PIV pioneer in Purdue. But this book is a really good purchase if you need to get into PIV. There's also an electronic version. If you have Springer access through your institution, you could download it for free. So when we think about all the different processing options, there's free, there's all the way from $0 to $50,000 types of options here, because the processing is really what you pay for a lot of times. Because when you buy the camera, you can get their appropriate grabbing sort of frame capturing software. You can maybe use a continuous laser pointer like Saad is using. You don't need to pulse it and do fancy, synchronization and all of that. So maybe all of that is quite easy. Processing, you need to kind of think of what am I going to do next? And a lot of times the processing, the nuance is like, does it give me the ability to like sort of tweak the raw image to maybe enhance contrast or something like that? Does it give me the ability to like, smooth the data after the processing is done? Does it give me the ability to like analyze further information like pressure from PIV, velocity field? Now can I analyze pressure from that? Right? So there's a lot of these is what you pay for. What I'm going to demo very shortly is Piv Lab. And so this is implemented in MATLAB. This is something I've been using for years now in my experimental fluids class. And I do PIV, I've done it in class. Have the students they'll have MATLAB through the institutional license. They install the software in MATLAB as an app even now. And then they're able to do things on the fly. We were able to sort of dive in to like see how good is this data, what would you change, et cetera, et cetera. There's also a free version called OpenPIV. It used to be implemented in both MATLAB and Python. They've gone to only do Python, and they also have a C++, I should say. So if folks that are somewhat familiar with Python, C++, this is a fantastic option. It's been used in a lot of papers. You don't need a license, commercial MATLAB license or anything like that. And then, MatPIV is something I used maybe 10 years ago. It was one of the first like non-commercial PIV software, but I mean, in quotes, if you need a MATLAB license, there's not a lot of pre and post processing options in this, and I don't think they support anymore. There's the JPIV, which is Java PIV. Again, some familiarity with Java is needed. Not sure if they still support this, but this has also been used in a lot of papers. And then in my lab, we've got this high-end software from LaVision called Davis. It's fairly popular in the sort of the fluids research community. This company and their software, they have quite a bit of uncertainty, quantifications done in their sort of calibration algorithms and all of that. So they've got a lot more robust, at least that's their claim. We've got hardware control. You can tie in cameras and lasers, computer, everything together within their program. You can process batches, which I found really useful. Before conferences, my students acquire like a terabyte of data and they go to sleep for like a week and the processing is done. And then you can do masking. We'll talk about this, pressure from PIV and so forth. And then I believe Eric might have access to KSI Insight. And again, that's another commercial software. The commercial software tends to be in the tens of thousands of dollars, if not more. And again, this has a lot of features of Davis. So what I'm going to do, and then I was going to mention this because I thought it would tie into what Saad was showing. There's a lot of movement for PIV for the people, because if we're reaching a point where we need to have more equity in who can do really good science without needing millions of dollars in grant money. And so there's this new paper that I landed on, which looks really interesting. I'd like to try it, but it's an app, Smart PIV. It's an experimental fluids paper. You can contact me if you want more details on it, but it looks really interesting. Years ago, I saw a SIGP meeting talk where they used I think they developed less than $1,000 total PIV system, something along the lines of probably a little bit more expensive than what Saad showed, but I wanted to kind of point them out as well. And then there's the Tomo PIV, which is 3-dimensional PIV with smartphones. So people are going really, leading this sort of revolution to try to make this methodology more accessible and equitable. Because smartphones are, even though they can be expensive, most people can't do without them. So can we use it for more than just taking pictures? Can we do more with them? So here's what I'm going to do. I'm going to jump on my other computer because the MATLAB's faster, but the Piv Lab is an open source MATLAB pro. So you have to have MATLAB. It needs the image processing toolbox, mainly the full license support, I think, that is needed for. But the homepage is really good. The developer, William Tilke, he's kept up with it constantly, all the time, really very good support. And the MATLAB toolbox is in the link below, and I'll give the slides to Brooke later. You're welcome to try it out on your own. So I'm going to jump, and it's going to kick me out, but I'm going to join in a moment.
Just as a reminder for folks who might have questions, you can post them to the Q&A box, which should be available on your Zoom palette. We're keeping an eye on that so we can answer questions. So Eric, you're going to do a Piv Lab demo as well, right?
Yeah, I was going to walk through some fish PIV, and maybe I'll focus a little bit more on the later stages of the processing and analysis. But I'll see what Arvind does.
Sounds good. And I think, can you guys hear me or no?
Yeah.
You can?
Yeah. Looks like you're on. You've got two.
I'm on two computers.
Yeah.
So,
looks like it's working.
So let me open MATLAB. So what I'm going to do is to show a data set that we acquired for my class just about a month ago, maybe.
So Arvind, you're still sharing from your PowerPoint at the moment on the first computer.
Okay. Yeah, I'll switch out here in a minute. Let's go here. All right. Now I should be sharing my MATLAB.
Yep.
Okay. So now I've installed Piv Lab, and so it's under apps. So it should be showing up here. Piv Lab right there. Can everybody see fine? No problem.
Yeah.
Great. Okay. So one thing I need to make sure is that it's sharing the sub windows from MATLAB. Are you able to see anything at all?
We just see the regular MATLAB interface at the
Yeah,
moment.
so let me share my whole screen because I think that's the only way to do this. Because MATLAB opens like a million screens after the fact. So desktop one, here we go. And let me minimize this. Okay, so far so good. Where did I go with my, oh, it's opening. All right. Okay, so it looks kind of like this, right? So it's really used, the graphic interface is really good. All right, so now what we're gonna do is load images. So the menu here sort of what is the sequence that is sort of recommended to follow. The calibration can be done before or after, and I'll get to it in a minute, but the others are sort of in the sequence. So it's kind of nice, you don't have to remember or have like a checklist or something like that. So you start a new session, load images. So I'm going to load some of these images here. So what I have is flow past the cylinder as it's being towed in water. Okay, it's being towed by a motor. It's got a couple of things on the ends of the cylinder. It's being dragged in the water. So I'm going to open 4 images, and then it's got this choice of, can you guys see my mouse pointer?
Yes.
Great. So it's got this choice of time resolved, which is the sequencing. So this is what it's going to compare. So this first bullet box or whatever is going to compare A to B, and then B to C and C to D. So it sort of double dips, let's say, in one image. So each pair will give you one vector field. So when you have a lot more like A, B, B, C, C, D, you get 3 vector fields. But if you did, for example, you did A, B, and C, D, you only get 2 vector fields. So it's not a deal breaker, but there's those options. So usually I don't use the reference. That's relative to a beginning image. We don't necessarily do that. It's far more specialized, I think. And then click import. It's going to load all this. And so you can kind of use this top thing to straddle between the images. So if you notice the cylinder, it's A to B is increasing time. So the cylinder is moving from the left to right. And it's a very small particle displacement. You could still see a lot of the particles are right there. They're not doing a whole lot. So this was recorded at 200 frames per second. And the speed of towing is 200 millimeter per second or 0.2 meter per second. So it's fairly slow. So let me move it here. And then the toggle here also gives you a way to kind of go from one to the next image pair. So this is, it calls frame as an image pair in the bottom. And so this is frame 2 and then frame 3. So it's just kind of going from B to B to B, can do the same thing from A to A to A, okay? So now we're gonna go to image settings. We're not acquiring images. This has been acquired by the camera program or something like that. So go to image settings, we do exclusions. This part is sometimes needed because we have solid objects that are creating a lot of issues in the flow. So like, for example, the cylinder is creating a very bright region And obviously there's no particle motion inside the cylinder. It's not porous or anything. So one idea is to say, I'm going to just select a region. I'm going to say, I'm going to just draw this box region, but I'm going to, process data. It's just going to do only processing in that data. So that's the region of interest. A more common idea would be to use like a mask. So you could draw masks for the current frame. And so the idea is they'll give you these crosshairs and you can draw, as close to what you want, like a polygonal sort of shape. And I'm doing it kind of on the fly, so it's not going to be as cylindrical, I guess. But anyway, and when you end the mask, it's very hard to tell, but there's a circle that it closes on the last dot. And so then this region, this red region is omitted from the processing altogether. So it's not going to worry about that region altogether. And so you'll have to apply these masks. It's just going to display, and then you have the choice to apply or clear. Do it again, clear all masks, all sorts of stuff. It also gives you the ability to load previously saved masks. You can have had a picture that you've saved. So I'm going to apply it. And so now it's going to not process that red region. And then here comes the pre-processing. So nearly all the PIV data we acquire the first time, it's not going to be really, really optimal. And I'll talk about a few best practices in just a few minutes. And so the programs give you an option to sort of tweak things a little. So this contrast limited, this sort of CLAHE routine, what it's going to do is it'll increase the contrast of the image. So you can't see it unless you apply and preview. So just kind of keep your eyes on the screen. And so it kind of pops the particles more so improving the contrast. So if you uncheck that and if you do a high pass, so this is sort of like what it's going to do is going to sharpen the image more. And so the intensity is dropped compared to the contrast limited that routine. But here it's going to make the particles more sharper. And so, this helps to sort of like improve make it more clear, the particle images. It's particularly useful when you have a lot of particles and you want to just kind of like pop them out more clearly without background. And then intensity capping is just going to go in and get rid of the background intensity to some extent, a threshold. And then there's these denoising filters. So there's a lot of different options you can play with. And the website, their sort of literature talks about this nuances of all of that. So I'm just going to leave it as the default here for what it's worth. And then I'm ready for my analysis. So now it's got the 3 different versions here. I believe DCC is the spatial sort of correlation. We don't want that. F50 is the one I just talked about, which is what we want. Ensemble correlation is used in micro PIV, not typically common use for a lot of normal scale PIV. But anyway, so the pass 1 is this sort of teal dotted window it's showing. So that's 64. It's a square window, 64 by 64 window. It's called interrogation area or interrogation window. And it's going to make the next window with a 50% overlap. So the next window is going to be separated by 32 pixels. And so it'll have a little bit of region of overlap. And then it's 64 by 64 all over the place. So that's one pass. And it's going to get an estimate of the velocity vector field. And then it's going to do a second pass with the first pass as a guess. So it sort of does a little bit more refinement. And so the second pass, I'm halving that window size. And like I said, smaller the window, you get more vectors because more windows, the windows are smaller relative to a larger window, so more vector fields. But anyway, here it's 32 by 32 pixels with a 50% overlap, which is half of 32 pixels. I leave these. You can read more in the Raffles book about these things, the subpixel, estimation and all of that. But it'll show you a first pass and then a second pass. And so this is the result that we get. And so what you see is this towing of the body. You get this large arrows. Those are large valleys of velocity. You get a vortex here and here, behind the advancing cylinder. You get a low speed wake here, practically not much going on down here, very much close to cylinders where things are happening. And then once you're happy with this, so you can play with this and analyze just one frame, it'll save time. And then you can analyze all the frames. And so it'll take some time. I'm just going to not do that because it's just a repeat. And then calibration, the idea is that at this point, it's going to have velocity in units of pixels per frame, which is not physical. And we want to convert it to meter per second and length in meters, like SI units, for instance. So we load an image. In this case, we put a yardstick in the field of view. And I'm going to pull up that image from my folder here. And it's going to pop that image. And then I select a known reference distance. And so this is like an inch, 14. I'm not going to do it super accurately because I don't want to take up Eric's time too much, but so 25 mm roughly. And then you want to pay attention, it's millimeter and milliseconds, a common error that a lot of people make at the first time, but it's 200 millimeter per second. So that's 1/200. So it's like 5 milliseconds. So I put that, and then you apply and it'll tell you the conversion. So now it says 1 pixel is what? That's like 0.2 millimeters or something like that, or 0.00021 meters. And 1 pixel per frame is about 4 centimeters per second, right? And so you apply that, and now it's going to give you all sort of real values. And now you can mess with the vector field. You can basically go in and say clear velocity, if there's velocity limits. So I need to get the velocity vector field data. There we go. Okay. So now you can go in and manually do surgery on it. So you can draw like a plot, or this is a scatter plot of all the velocities independent of the location. This is how much variation there is in the data set. And X velocity is in the X-axis, Y in the Y-axis. And so I go in and click that. It's going to go in and remove everything else that is outside of that box. So if you notice, I didn't pick a good limit because it removed a lot of the useful stuff, the higher speed flow. So this is something you don't want to necessarily do unless it's a very uni sort of velocity scale. Obviously here, there's large velocity here, very slow velocity here. It's hard to kind of narrow down like a nice region. So I'll do undo all the validations, and then you can apply some sort of standard deviation and it'll remove. So what it does, you can also ask it to apply, it will remove anything that's above that threshold of 8 times the standard deviation of the vector field, the velocity value. And this is a median value instead of a standard deviation. You could do notch filters. But anyway, so if you do this, what it's doing, so I might have to do something aggressive to show a better example. Oh wait, that's the wrong way. Okay, so what I did is I said, I only tolerate 2 standard deviations and everything else, I'm going to remove it. What Piv Lab does, it'll replace it with artificially interpolated calculated vectors. You can change the color. The default is red. So all these red vectors here are not real, are from the algorithm, not the original sort of cross correlation outcome. It's sort of artificially, I guess, altered. So most people will do some level of post-processing and report it in the paper. So it's not a bad idea to do post-processing. It's just that you don't want the entire, like half of this window to be all red vectors, because that looks like, you're doctoring the data beyond a point. And then, I think at this point, I probably should let Eric take over, like taking up too much time. But the next set of steps that Eric's going to talk about, I guess, will be all the different displays. So Eric, is this a good stopping point for you? Do you want me to go into anything else?
Oh, this is great. I can.
Okay, perfect. All right, let me stop sharing. Anytime I need to stop sharing, I can't find my window. Oh, there we go. Stop share.
All right, so let's see. So I'll come in. Any questions for Arvind before we move on? Not seeing anything. So yeah, so I will start here, and I'm going to start by sharing this window. So what I'm going to attempt to do is a very quick version of the analysis we did in this paper a few years ago using Pib Lab, which is actually not the software we used for it, but I think it will illustrate the point. So this was a paper out in 2018. This is Tyler Wise. This was actually an undergraduate senior thesis. So a lot of this analysis is possible to do with undergrads, and kind of fun. And there was a postdoc, Margot Schwalbe was a postdoc at that point. She's now a professor at Lake Forest College. And just to orient you before I start showing you PIV videos, this is the setup. So we have our, can you all see my mouse? Anyone see my mouse? Can people hear me? Okay, so we have our fish swimming in a flow tank. There's the flow coming against it. We have two lasers set up. There's one coming in from the side and one actually reflecting off a mirror behind the fish, illuminating from behind. And the point of doing this is that we don't have shadow. And then we're videoing the whole thing from below with a high-speed camera. So this is definitely one of the sort of expensive setups. What I will note is that, when I bought all this equipment about 10 years ago, the prices of all of these things are between a third and a half what I paid right then. It's not to say they're cheap yet, but all these prices, even for fancy equipment, is going down. And a lot of what we've done here, you can now do with much less expensive equipment. So, okay, so now stop the PowerPoint. Piv Lab. Okay, so I'm kind of at the point where Arvind finished off, except that I have a fish. So what we're seeing here is a fish from below illuminated by the laser. So the one laser is coming in from the top of the screen towards the bottom, and you can kind of see a slightly darker where it is shadowed. And then another laser is coming in from behind, illuminating this side. And you can see all these particles here. This is maybe not the world's best PIV image. It's a little overexposed. You can see all these particles are getting up to the maximum intensity, and ideally we wouldn't have that. It sort of just hit the maximum, but we wouldn't have these bright white spots there. But PIV is really robust. It works very well in all sorts of less than ideal conditions, and it works really well in these particular videos, which is why we showed them. So let's see. Okay, so let's show the vectors again. So the first thing I'm going to do is a bit of this validation. And what you'll see here that's typical, or let me just step through. So if we step through the images, you can see the fish's tail is moving towards the top of the screen. I've processed all the frames, but what I haven't done is the manual masking that Arvind showed. And particularly when you have an animal moving around, it's really time consuming to do that. And it's kind of hard to do automatically as well, because you see we have this dark region that's part of the fish, but we also have this really bright region that's part of the fish. But the algorithm here, it just runs across everything. So we have vectors that are on the fish body, we have vectors that are in the flow. One thing that Piv Lab does, if my zoom wasn't in the way, is in the post-processing, this image-based validation actually works fairly nicely in these kinds of situations. And in particular, this low contrast filter, in this case, works pretty well. What this does is it looks for regions where there's not a lot of change in contrast. So when we have good particle images, we'll have dark regions and we'll have bright regions. But on the fish, we might have all bright regions or all dark regions. So if we apply this here, what you'll see is now I've replaced a lot of the vectors on the fish with these orange vectors. So that can be helpful. Piv Lab really likes to interpolate the missing data. If I were processing this on my own, I wouldn't interpolate the missing data because then it just wouldn't be there. But you can decide how you want to handle that. But for the moment, I'm going to interpolate it. This velocity-based validation is also useful. And so here, Arvind showed this as well. Again, we're showing all of the u velocities of all of the vectors on the x-axis and all the v velocities of all the vectors on the y-axis. And you'll see in this case, I've got a cluster over here near 0, 0, and a cluster over here that has some positive u velocity and positive negative v's. And in this kind of case where you have something swimming against a flow, this region is the body, basically. That's the fish because it's stationary in the flow field. And this region over here is what we're interested in. So in this case, if we select this whole region, what we're going to do is eliminate even more of these vectors that are on the fish body. So that's a really important thing to do to make sure you're not accidentally including things that relate to the movement of the fish rather than the movement of the flow. And you'll see here this is the fish's tail here. Out here, it gets really hard to do anything automatic. And so you just have to be careful about your analysis there. So, okay, so the next thing we typically do in these kinds of cases is we want to subtract the background flow. So you'll see the fish is swimming against a flow, all those vectors are moving from left to right. It's kind of hard to see what's going on. And that's actually in this derived parameters in Piv Lab. We can subtract the mean flow. So I'm going to grab the mean U there, apply to current frame. Now you'll see it almost looks like a lot of the vectors have gone away. That's because I've taken that mean left-right flow and I've gotten rid of it. So now we're seeing kind of zero flow in here. But now what this lets us see in a moment is the wake. So to be able to see the wake a little better, I'm going to modify the plot appearance, and I am going to increase the scale of those vectors. And so now I have much longer vectors in the wake. And now you can start to see, going back a little bit to what Matt showed at the beginning, these sort of counter-rotating vortices in the wake of the fish. And let's see. So now what we can do, typically when you see these plots published in a paper or something like that, you'll see the vectors sometimes, but we'll often move on to vorticity pretty quickly. And the reason we're interested in vorticity is really a signature that something has produced a force in the fluid. It's a complicated interaction, but we look at vortices in a lot of cases because we're interested in forces. So let's go in and figure out our vorticity. And so we'll go into these derived parameters. We'll derive our vorticity. Let's apply that to the current screen frame. And so now we have our vectors and our vorticity. The vorticity is the color in the background. So here we have clockwise rotation, counterclockwise rotation. And you can see this nice vortex pair here with a jet in between them. You'll notice, though, that the fish has disappeared. So now we have no idea where the fish is anymore. So this is one of the reasons why we need to be sort of careful to eliminate those vectors that correspond to the fish before we start doing this processing. So I would be very distrustful of our vorticity estimates in this region because I know the fish's tail is there. And so we're taking these velocity derivatives across a solid object, which doesn't make any sense. But out here, I've chosen this frame. So this vortex is fully shed, it's in the wake. The tail isn't there. So having done this, I said we look at vorticity because we're interested in forces. Typically, the next step we need to do is quantify the strength of our vortice. And we do this, even though we show it with the way I usually suggest to do this is using a parameter called circulation, which is a measure of the total vorticity and Piv Lab can do this for us. We'll go to extractions and we'll extract parameters from polyline. And in fact, what I'm going to show you is what it calls circle series here. So vortex, well, let me just show you and then I'll explain what it's showing. So I'm going to draw my circle. And so I'll click on the center of this vortex just by eye. It's not too sensitive to where the center is. And then I'll click somewhere out further away. And that second click is a radius. And so what it's going to do here is it's going to draw a set of increasingly larger circles starting at that center point. And some of these over here are going to intersect the tail. So you need to be careful. And then I'm going to plot the data. So do you all see this other window? Another window popped up for me.
No, it's
okay.
Probably right. Because the MATLAB sharing. Yeah.
Yep. So let me just share the whole of that desktop if I can figure out how to do that. There we go. So, right. Now, we're good here. So you should see another plot with kind of a diagonal line going down, right? Okay. So what this is showing us is the circulation. So it's the integral of the velocity stepping around that circle. But that also represents the sum of the total vorticity in that region. You can work out the math later. And we're stepping outwards. Our x-axis here is increasing radius. And our y-axis here is this integral of the velocity around the circle. And what we'll see is it steps out and then it kind of levels off and then it steps out. And so if this were a single isolated vortex, what would happen typically is it would step out and then it would level off. And what we'd see is that there's kind of a core region and then an outside of the vortex region. Now, this isn't a single isolated vortex. It's a piece of a wake with connected vortex pairs. And so there isn't this clear distinction between the core and the outer portion in the same way. But typically, I would choose a point where we sort of get this inflection point. And you'll see it's now highlighted this radius here. And I can look at that and say, is most of the vorticity contained in that radius? And it looks all right. Again, this is going to be a judgment call, but this gives me a measurement. It's a very tiny number here. It says 0.0045731 meters squared per second. So this is an integral velocity, meters per second, over a distance or times a distance. So we get meters squared per second, that's our circulation. And so if we want to know the impulse that produced this vortex pair, we could do the circulation in this vortex core, we could do the circulation in that vortex core, and then it's related to the area of the vortex pair. So this is a slice through something roughly circular. So we take the distance between the two, and that would give us an estimate of the force that produced those. Before we go too much further, though, I will say that since this is kind of a judgment call on what the vortex core actually is, what I tend to suggest, and what we did in part of this paper, is that you can actually do it in a slightly different way. What you can do, if you go to this polyline option, you can draw a line, and instead of going through the vortex cores, what I'm going to do is I'm going to draw a line through the center of this vortex pair. And if this were an isolated vortex pair and not a member of a connected wake, the velocities out on the edge would go totally to zero. And so I can assess the tangent velocity along this line, plot that data. And so it shows me the velocity parallel to this line along the length, but conveniently down here, it has this integral of that. And it turns out in a lot of cases, that's a better estimate of the circulation of these vortices in the wake rather than trying to zoom into the core and use this judgment call. So what I get by doing it this way is 0.0049, which is pretty close to my judgment call from before, but it requires less judgment because I can see that the velocity has gone mostly to zero at one end, that's up here, it's gone mostly to zero down here, and somewhere in the middle, and here, that's where the velocity is highest. So with those circulation measurements and the distance between the vortices, we could estimate the impulse required to generate this vortex pair, and we can use that. So impulse is force multiplied by time. We divide that by the tail beat period. We can start to estimate how much force this fish is producing every tail. So let's see. So that is a really quick run through of the sort of analysis we did in that paper. I want to show you a few more things we can do back with PowerPoint that we didn't do in that paper. Okay, so these are my notes for what I was doing. And so a few more things that we can do from these kinds of measurements. One thing that's becoming more common these days are different algorithms for estimating the pressure field from the velocity field. And the algorithm I'm familiar with was developed by John DeBury and colleagues in 2014, and then really validated by Kelsey Lucas in 2017 for fish type flows. And then I'll just show a few quick pretty images of Lagrangian coherent structures, which is a way of sort of assessing what parts of the wake are important. So here, so for pressure estimation, one of the really crucial things about doing these sort of pressure estimations is you need a full field flow. So you need not to have shadows in your flow, because what we're going to do is we're going to integrate from the edges of the flow to our body in the center. And if we have shadows, then there's some edges that we don't have data for, and that makes it much harder to deal with. It's not impossible, but it's far better if you have a full field of flow. And then you also need a fairly wide field of view. So if you really are zoomed in close to the fish or whatever you're interested in, it's not going to work so well because we have to assume that the pressure at the edges of our field is zero or is ambient. And so the steps to do this is you do PIV, just like we've been showing. In this case, you really do need to outline the body because we're interested in how the pressure impacts the body itself. And then John Dabiri has made this Queen 2 processing code available. It runs in MATLAB as well. And it's fairly straightforward once you've done these two steps. So here's our full field flow, like I showed before. In this case, the fish is a little more centered, which is good. You can see we have, so there is a shadow here, but since we have the second laser, there's enough particle information there that we can still get the vectors in that point. And similarly, there is a shadow here, but again, with two lasers, we were able to keep that usable. Then Tyler went through and sort of manually traced around the body here. These vectors are just the normal vectors from this tracing. And he had to do that for every frame we were interested in, which took quite a while, but he did. And again, it's hard to do automatically because of the differences between, there's a lot of contrast here, there's a lot of contrast here, standard edge detection doesn't work very well. So if anybody knows an automatic way of doing this, it would be nice. But then from here, we can run this algorithm and estimate the pressure. So this is the pressure in pascals, and we can integrate that up to the surface of the body, run this. So we can see high pressures developing along the tail. We can use those to estimate the forces on the tail, the propulsive forces. We also get some low pressures up here along the body. So there does seem to be some suction forces involved as well. So that can be really useful for understanding the force distribution along the body. And so finally, they asked me to mention Lagrangian coherent structures. This is not something I've actually done before. So I pulled out some images from an older paper now, but some of the sort of fishier or the more fish-like examples I could find. This is a plate flapping in flow. The black and white here are positive and negative vorticity. And what Melissa Green and her colleagues have done here is they've used these finite time Lyapunov exponents. What it does is it's kind of a way of looking at regions of the flow that stick together. And so you take a section of the flow and you step at the velocity at that point. And then you keep on stepping, and then you choose another point and you step at the velocity at that point and you keep on stepping. You look for regions where things tend to diverge, and you can step forward in time and backward in time. And so these colored regions are the regions where things tend to diverge. And so if we track the forward and backward time, we end up with this region here. And then inside there is a region where things tend to stay together. And so what this helps us do is identify coherent structures within the flow, regions that typically stay together. In this case, what we're looking at, what we find then is that we're identifying these linked vortices in the wake. And so this can provide a little bit more of an objective way of identifying the regions in the wake that we need to process if we want to understand some of these production. So I think that was all I was going to say.
I think we deserve a pat on the back for staying on schedule. I'm really impressed.
Thank you so much for that. It was really interesting. See if, does anybody have a question to start things going from our attendees?
I mean, I can just say I enjoyed everybody's presentations. It was fun. It was fun to view.
Just to remind everybody, I posted in the chat that this will be posted online via the IOMM webpage. And I also linked all of the presenters' personal lab web pages where you can also learn more.
We do have one question in the Q&A.
Yeah, so I could probably take that. I think they start talking about it, but I don't think the textbook goes into a whole lot of tomo-PIV as far as I remember. Right now, I can tell you in a minute. Yeah, there's a chapter on 3D PIV, but I think it's still not evolved to the point where one could write a whole book in it, maybe. But the chapter is really useful. It builds off of a reference paper that is well cited by Elsinga, Elsinga, and tomo-PIV, and so that's where this sort of builds on that. So
yeah. If I can chime in on a slight tangent there, I'm not sure what the purpose of the question is, but my suggestion in general is to avoid trying to do volumetric techniques unless you really, really need that sort of information. There's getting the densities correct, getting the velocities correct is orders of magnitude more challenging for volumetric techniques, and just much harder to know whether you're doing it right. So, and oftentimes you can get most of the important information out of a 2D trace than you can out of the volumetric work and save yourself a lot of heartache. I see some nods, but do the other presenters agree with me there?
I think I do. I think definitely, like, if you want to understand the fluid dynamics for new system, it's much more recommended to start with 2D first to really kind of get a flavor, like a feel for how complex the flow field is. And with 3D, I think it's always, like Eric, you mentioned, it's absolutely true. It's hard to know whether that data is good enough, especially with a lot of the post-processing. It's easy to sort of kind of make something look okay, but there's some serious issues with the data. So I think 3D, I think, is more useful when you have a better idea of the system, the physics of the system. So at least to where you can expect, what to expect. I think it's hard to go in, a new system just 3D with absolutely no a priori understanding of the flow physics, because there's no way to tell what you're getting is, does it even make sense? Because you could be interpreting something that is affected by artifacts rather than actual physics. So it's extremely time consuming. And when you have bodies in motion, that's, what is it, quadruple the work with 4 cameras? I have collaborators that do, tomo-PIV on Krell, and it's you can't do statistics, so easily because it takes so long to process the data that, you can't do multiple, individuals and get a lot of statistical, sort of outcomes from it, not without having an army of people at the very least. Yeah. So, but Saad, it was nice to see your lab. I really enjoyed that.
Yeah, that was great.
Yeah.
Thank you. I think, yeah, this was helpful. I actually told some of my students to watch some of the analysis stuff as well, because I think there are levels of how detailed you do the experiments, and
right.
Yeah, as the computational analysis part, we were putting together kind of a graph for posting on archive soon about how to get started on this. But then always, if you want to improve, like going up to PI, doing tomo-PIV and stuff, sky's the limit. But I think what you mentioned was very good. Sometimes just doing the quick and early step is helpful, especially if sometimes you want to do things in the field or outside the lab. Simpler the better. Most animals do not cooperate with you, which, yeah. So getting started is, I think, half of the battle, and then you can always continue to refine and improve.
Right. Right. Absolutely.
This was a lot of fun.
Yeah, it was a lot of fun. Yeah. Well, thanks, guys. Thanks, everybody. Yeah, it was fun.
You, everybody.
Yeah.
Thanks to
Thank you all very much.
All right. Bye-bye.
Take care. Have a good weekend. Bye.
Bye.